What if the enemy you're fighting at this very moment is neither alive nor dead? Viruses replicate. Yet by many definitions, they fall outside the realm of life. Scientists have spent centuries trying to define life. NASA, for instance, defines it this way: a system that self-replicates, metabolizes, and evolves is life. But the boundaries are blurred. In 2010, scientists rebooted a cell with chemically synthesized DNA. Today, we confront a fundamental question. What is life? And what are we, ourselves? By the time you finish this video, you'll realize the boundary between life and non-life is far more ambiguous than you ever imagined. Chapter 1. Existence on the Boundary. Let's look at viruses. They replicate, yet they can do nothing on their own. Are viruses alive? Many definitions exclude them from life, though some researchers view them as a form of life. The conclusion is far from unanimous. Viruses don't metabolize. They cannot generate their own energy. Only after invading a host cell and hijacking its machinery can they replicate. This is a remarkable strategy. Viruses are the ultimate minimalists. They carry only the bare minimum genetic information and borrow everything else. In 1935, scientist Wendell Stanley succeeded in crystallizing the tobacco mosaic virus. He crystallized life, like salt or sugar. Later, understanding progressed that this was a particle containing RNA. And here's where a crucial question emerges. If life can be crystallized, what distinguishes life from matter? Is a virus a thing, or is it a living being? But in recent years, giant viruses possessing some metabolic functions have been discovered, shaking even this definition. Scientists are still searching for answers. Viruses are entities that shake the very definition of life. But this is only the beginning. So can we artificially create life? Let's explore that answer. Chapter 2. Life that is created. In 2010, scientist Craig Venter stunned the world. He transplanted a chemically synthesized DNA genome into an existing bacterial cell, creating a self-replicating cell driven by that genome. How? He designed a genetic sequence on a computer and synthesized it chemically. Then he transplanted that artificial DNA into the empty shell of another bacterium. That cell divided. It replicated. What matters is that the synthetic DNA hijacked the existing cellular machinery and drove the cell. He didn't chemically synthesize an entire cell from scratch. Let's think about what this means. Many researchers consider life a phenomenon explainable by physics and chemistry. Yet how life emerged from non-life remains unresolved, with multiple competing hypotheses. Research then progressed to how minimal can you go while maintaining life, narrowing it down to 473 genes in minimal genome cells. Even so, essential genes of unknown function remain, and the minimum requirements for life are still not fully understood. Scientists have hypotheses about life's origins. The RNA world hypothesis, proposing a stage billions of years ago when RNA served both genetic and catalytic roles, is one leading candidate, though timelines and pathways remain unconfirmed. The most poetic and moving scientific fact is that many of our atoms, carbon, oxygen, iron, were forged in stars and supernovae. Meanwhile, hydrogen primarily originates from the Big Bang. Think about this for a moment. If life consists of chemical reactions, does a true boundary between life and non-life even exist? Isn't it a continuous gradient rather than a clear line? This is also a philosophical question. To define life is to define ourselves. But here the story takes an unexpected turn. So, what does this discovery tell us about our own existence? Let's explore. Chapter 3. What are we? So what about us? The human body consists of approximately 37 trillion cells. Yet, in terms of numbers, human cells and microbes are roughly equal, or microbes may even slightly outnumber them. What does this mean? Bacteria, viruses, fungi live symbiotically in our bodies. Without them, we cannot survive. Gut bacteria digest food, produce vitamins, and regulate the immune system. They are part of us. Yet they themselves are independent life forms. This brings a profound insight. The very concept of individuality may be an illusion. We are communities of countless lives. Even more remarkably, the mitochondria inside our cells were once independent bacteria. Over 1 billion to 2 billion years ago, they were incorporated into other cells, forming a symbiotic relationship. In other words, evolution progresses through cooperation. Not just competition, symbiosis has made life complex. Let's think about what this means. If we are assemblages of countless lives, what does I refer to? Where is the boundary? Or does no boundary exist at all? Finally, I want to ask you, if life is merely a succession of chemical reactions, where do our consciousness and emotions come from? And should we grant the same dignity to artificially created life as we do to ourselves? Science alone cannot answer these questions, but continuing to ask them moves us forward. So let's organize what has emerged from this exploration. Chapter 4: The World Revealed. Now, today we've deeply explored the definition of life. What we've discovered is that the boundary between life and non-life is far more ambiguous than we thought. Viruses exist between life and death. Artificial cells run on chemically synthesized DNA, and we ourselves are communities of countless lives. Let me share the insights gained from today's exploration. First, the boundary between life and non-life is not a clear line but a continuous gradient. Viruses can be crystallized, yet also replicate. Giant viruses even possess some metabolic functions. Where does life begin? The definition continues to shift. Second, life's origins and minimum requirements are still not fully understood. The RNA world hypothesis is leading but not confirmed. Even minimal genome cells retain essential genes of unknown function. We still don't fully understand the essence of life. Third, we are not solitary individuals, but ecosystems where countless lives coexist. Gut bacteria, mitochondria, all cooperate to constitute me. These three insights fundamentally transform our view of life. We are not isolated beings, but part of the universe's chemical reactions, communities with countless lives. The more we explore life's definition, the more ambiguous its boundaries become. But that's not uncertainty, it's richness. Life is far more diverse, flexible, and creative than we imagine. And I feel a sense of awe. A succession of chemical reactions generates consciousness, feels beauty, and poses questions. Isn't this miraculous? After finishing this video, when you return to daily life, I want you to be aware for just a moment. Inside your body, at this very moment, 37 trillion cells are cooperating. Gut bacteria break down food, mitochondria produce energy, immune cells battle invaders. And the next time you catch a cold or look in a mirror, think about this. What is I? Where does me end and other begin? Share your insights in the comments. Your questions become the force that propels this channel forward. Thank you for joining me on this intellectual journey to the very end. The question of what life is the question of what we ourselves are. On this channel, we continue exploring the universe, consciousness, and the essence of existence. If this exploration resonated with you, please subscribe and hit the notification bell. Next time, I have questions prepared that will shake your worldview. Until then,